In the last lecture, we covered some of the characteristics of the plant immune system and how plants can respond to viral, bacterial, and fungal pathogens. In this lecture, we'll focus on ways plants defend themselves from herbivores, as well as how plants allocate resources for defense. Plants have evolved mechanical, chemical, developmental, and even ecological means to protect themselves from herbivores. Milkweed is an example of a plant that has developed both mechanical and chemical defenses against herbivores. The leaves of the milkweed species are covered with dense hairs, as many as 3,000 hairs per square centimeter. An insect crawling on a leaf must make its way past these hairs before beginning to feed. Milkweed has another defense in its leaves, a sticky liquid called latex. This latex is found in extracellular channels and is under pressure in an intact leaf. This pressure then causes the latex to flow out of the leaf and stick to anything it comes in contact with when the leaf is damaged. A feeding insect is in danger of having its tiny mouth parts glued together if the latex comes into contact with it. The monarch caterpillar has evolved to be a specialist herbivore and has developed methods to get around the latex defense of milkweeds. Smaller monarch caterpillars will cut trenches around the area they intend to eat to prevent the latex from flowing into that area. Larger caterpillars are able to avoid the latex by severing the midvein of the leaf. This is the central vein that runs the length of the leaf. Severing this vein relieves the pressure in the latex canal system downstream of the cut, allowing the caterpillars to feed in this area. Milkweed latex is toxic in addition to being sticky. It contains high concentrations of cardenolides, which are steroid compounds that cause heart arrests in animals. However, monarch caterpillars do not metabolize the cardenolides as they consume. Instead, they sequester them within specific regions of their bodies. This storage provides the monarchs with a chemical defense against their own predators because they become highly toxic to birds and animals that would otherwise eat them. Hairs are a common mechanical defense plants employ against herbivores. Many times these hairs are also armed with chemical irritants. Example of these hairs can be seen on the leaves of this nettle and this Arabidopsis. Some plants have prickles, spines, or thorns like this beach palm. Grasses and other plants also have a mineral defense consisting of silica plates within their epidermal cells. The plates function as a mechanical defense because they wear down insect mouthparts, so the insects feed less efficiently and grow more slowly. Plants use a wide range of chemicals for protection. Some plants produce nitrogen-bearing compounds called alkaloids. These compounds typically taste quite bitter and damage the nervous system of animals. Alkaloids include compounds like nicotine, caffeine, morphine, theobromine, quinine, strychnine, and atropine. Because alkaloids contain nitrogen, which is often a limiting element that plants need, the production of alkaloids is a costly defense. This table is a summary of major plant chemicals that are used in human medicine. Visacrine is an alkaloid extracted from the Madagascar periwinkle and was one of the first chemotherapy drugs as it inhibits microtubule polymerization and thus prevents cell division. It and other alkaloids do not harm the plant producing them because they're often stored in extracellular canals or otherwise sequestered. Terpenes or terpenoids are a second group of defensive compounds. Unlike alkaloids, terpenes do not contain nitrogen. Small terpenes are volatile and vaporize easily. These small terpenes make up many of the essential oils and distinctive smells associated with plants. Many of these odors, like methanol, may be pleasant to us, but they serve as feeding deterrents to other mammals such as squirrels. The growth, development, and metabolism of both fungi and insects are also negatively affected by terpenes. For example, phytoesterics are plant-produced replicas of hormones that induce the molting in insects. Exposure to these compounds keep insect populations in check by causing insects to molt prematurely. Taxodiene, which is a precursor for the chemotherapy drug Taxol, is a terpene that was first extracted from the bark of the Pacific yew tree. The third main class of defensive compounds are the phenols. These are exemplified by the tannins, which are found widely in plant tissues. Tannins bind with proteins and reduce their digestibility. Tannins are stored in vacuoles by plants so that only when the cell is damaged do the tannins come into contact with the proteins in the cytoplasm. Plants will often store high levels of tannins in unripe fruit to prevent their consumption before the seeds within them are mature. Some of the chemical defenses used by plants are stored in separate compartments from the enzymes that activate them. When you bite into a plant belonging to the cabbage family, the chemicals stored in the vacuole commingle with the enzymes in the cytosol. The enzymes then catalyze the production of mustard oils from these compounds. Mustard oils give cabbage and its relatives their distinctive smell. They also serve as deterrents because they interfere with insect growth. The two final chemical defenses found in plants that I want to discuss are protein-based. The same 20 amino acids are used by plants and animals to construct proteins. However, plants produce some additional amino acids called non-protein amino acids. Plants do not use these additional amino acids themselves, but the herbivores that consume the plants can run into trouble. There are a few ways that non-protein amino acids can have a direct toxic effect on herbivores. These amino acids can be misincorporated into proteins and disrupt their function. Some non-protein amino acids obstruct primary metabolism, and others interfere with insect neurological processes. A second protein-based defense is the production of antidigestive proteins called protease inhibitors. The protease inhibitors work to disrupt the enzymatic activity of the digestive enzymes present in the stomach of the herbivore by binding to the active site of these enzymes. These enzymes can no longer properly digest the plant material and thus reduce the growth of the herbivore. Though not as common as mechanical or chemical defenses, a small number of plants have evolved ecological defenses against herbivory. In exchange for shelter and/or food, these types of plants are able to use animals as bodyguards. In addition to the nectar found in flowers, many plants also produce nectar in glands located on their leaves. These nectar-producing regions attract ants, which then move over the whole plant in search of more nectar. When the ants encounter either eggs or larvae of other insects, they'll consume these as well. The value of this relationship can be seen easily because when the ants are prevented from patrolling certain branches, those branches suffer higher rates of herbivore damage. The group of plants known as the grasses are well adapted to cope with grazing animals. Their stems exhibit limited internodal elongation, and their shoot apical meristem is found very close to the ground until it is time to flower. Additionally, the leaves of grasses have zones of persistent cell division and cell elongation at their bases. This means that as animals come by and graze the tops of leaves, the bottom of the leaf can continue to grow and quickly replace the damaged blade. Plants invest substantial resources in defense that might otherwise be used for growth and reproduction. The benefits, therefore, of defense must be large given the considerable costs. In nature, the balance between cost and benefit is achieved in several ways. When a threat is constant, plants use defenses that are always present, known as constitutive defenses. When a threat is only occasionally encountered, a plant may mount an inducible defense. This is a defense that is activated only when the plant senses a threat. Inducible defenses can either up the level of constitutive response or trigger a new type of defense. Plants are able to sense and respond to herbivores. Damage caused by herbivores triggers the synthesis of jasmonic acid. This signal is transmitted through the phloem, and exposure to jasmonic acid leads to the expression of defensive genes, even in parts of the plant not directly affected by herbivores. These genes increase the production of chemical defenses and stimulate the mechanical reinforcement of cell walls. An example of constitutive and inducible defense can be seen in coyote tobacco. This plant is attacked by a variety of both generalist and specialist herbivores, but which species are damaging varies from year to year. So the plant must either produce costly defenses for herbivores that may not be there, or be able to identify and respond to specific threats. A major component of its defense is the production of the alkaloid nicotine. This defense is metabolically expensive because it contains nitrogen, but is effective against many generalist herbivores. Coyote tobacco plants that have not been damaged by herbivores produce only low levels of nicotine as a constitutive defense. However, once a generalist herbivore starts to attack the plant, nicotine concentrations rise quite a bit in response, in an inducible response. However, coyote tobacco is also attacked by a specialist herbivore, the tobacco hornworm caterpillar. These caterpillars are able to consume leaves with high levels of nicotine because they have evolved the ability to sequester and secrete nicotine. How does the plant then protect itself against these caterpillars? In a kind of a neat trick, coyote tobacco is able to recognize specific chemicals in the saliva of the caterpillar. Instead of producing more nicotine, which would just be a waste of nitrogen against this herbivore, it produces volatile signals that attract other insects that will attack the eggs and larvae of the caterpillar. Plants respond to herbivore damage by producing chemicals that make their tissues less palatable. Field experiments have suggested that plants neighboring those that are damaged by herbivores will also increase their production of defensive chemicals, whereas plants further away will not. Scientists hypothesize that volatile defensive chemicals released from attacked plants can trigger a defensive response in neighboring plants. To show that this happens, a set of plants was placed downwind of damaged plants, which had leaves torn to simulate herbivore damage. A set of control plants was placed downwind of an empty chamber. The concentrations of defensive compounds in all sets of the plants were measured and compared 52 hours after the initial damage. Elevated levels of defensive compounds were found in both the damaged plants and the undamaged plants downwind of them as compared to control plants. Follow-up work showed that the volatile chemicals reduced from the damaged plants attract predatory animals that feed on the herbivores, parasitize their eggs, or both. This is like the coyote tobacco's response to the tobacco hornworm caterpillar. Plant species vary significantly in the extent to which they allocate resources to defense. Because defenses are costly to produce, you might assume that plants growing in nutrient-rich habitats would be the best defended. However, the counterargument is that plants growing in nutrient-poor soils might invest heavily in defense because they cannot afford to replace tissues lost to herbivores. These plants would grow relatively slowly because their resources are being used to build defenses rather than for growth. Plants in nutrient-rich habitats would put growth over defense because they could readily replace lost or damaged tissues. This situation is an example of an evolutionary trade-off between growth and defense. A potential example of this trade-off between growth and defense was studied in the Amazon rainforest. Plant species on nutrient-rich clay soils were compared with closely related species found on nutrient-poor sandy soils. Researchers found that species from the nutrient-rich clay soils invested less heavily in defenses than did species from the sandy soils. Both species grew best in their native habitats when under the threat of herbivory. However, the clay soil species grew much faster than the sandy soil species when the plants were protected from herbivory. Plants are not often just under attack from a single pathogen, herbivore, or competitor. With multiple simultaneous attacks, plants often respond with a trade-off, since they have limited amount of energy available for defense. A plant attacked by a pathogen may respond less vigorously to a subsequent herbivore attack. Studies indicate that crosstalk between the signaling pathways for responding to pathogens versus herbivores is responsible for their reduced ability to respond subsequently to herbivores. This allows the plant to invest more of its limited energy into fighting off a potentially deadly pathogen. Despite the large proportion of resources plants allocate to defense, insects and other pathogens still consume or destroy a significant portion of plant growth. They affect not only wild plants, but the plants we also choose to cultivate. This means that herbivores and pathogens are a major concern for agriculture. A common practice in modern agriculture is to plant large plots with single species, often a single genetically uniform variety. The result is that herbivores and pathogens are not slowed by low target density or genetic diversity in terms of defense. This can lead to losses in yield, and protecting these crops from damage becomes a high priority. Pesticides and herbicides are important contributors to the increased crop yield brought about by modern agriculture. However, in addition to the toxic effects of these additives, widespread chemical treatments provide a strong selective force for the evolution of resistance. In other words, by applying pesticides, we select for organisms that are resistant to those chemicals. Then, as pests become resistant to the pesticide, One response is to apply more or stronger chemicals, which increases the selective pressure. One consequence of the increased chemical application is the death of the pest's natural predators. The application of increasing amounts of pesticides to rice paddies in South Asia in the 1980s was not met with a decrease in crop losses. This dangerous course was reversed when farmers instead started devoting substantial effort into determining what pests were actually present, whether or not they were at damaging levels, and whether or not the pests had natural predators in the region. Only after understanding the ecology of the farm were the pests were removed. First, the pests were removed via mechanical means, then through biological control such as predators, and then only as a last resort, industrial pesticides were applied in some cases. This approach is called integrated pest management and is designed to minimize the evolution of resistance to pesticides. Crop breeding is a powerful way to alter the genetic makeup of cultivated species to improve defenses against pathogens and herbivores. Traditionally, this involves selecting and breeding for favorable traits or to introduce specific genes into a population. However, because it relies on sexual reproduction, crop breeding is limited to the genetic diversity that exists within interbreeding varieties. Rhizobium radiobacter, the bacterium that causes crown gall disease, has become a powerful tool which allows scientists to introduce specific genes found in distantly related organisms into cultivated species. The introduction of chemical defenses from a bacterium into crop plants was one of the first commercial uses of this technology, and we'll talk about this more on the next slide. Bacillus thuringiensis, or Bt, is a bacterium found in the soil that is toxic to insects. Since the 1920s, it has been used as a method to control insect outbreaks around agricultural crops. Early methods applied the toxic spores or the toxins directly to the plants in the field. Then in the 1980s, the genes that encode for the toxin in Bt were inserted into tobacco, where their expression by the plant provided protection from insects. Now, plants that have been modified to carry the Bt genes are some of the most widely planted genetically modified crops. While the introduction of Bt crops has reduced the application of pesticides to fields, the constant exposure of the pest populations to the Bt toxins in the Bt crops has increased the probability that these pests will evolve resistance to the toxin. This is of great concern to organics farmers who still use the application of Bt spores to control for insects rather than Bt crops themselves. To combat the evolution of resistance, farmers in the U.S. are required to plant refuges in a fraction of their fields. These refuges contain plants that do not express the Bt genes, and they serve as food for insects that are not resistant to Bt. By maintaining non-resistant insects in the population, the evolution of resistance is slowed as they interbreed with insects that are feeding on the Bt crops. The ultimate efficacy of this practice has been questioned due to response of Bt-resistant pests and the need to apply increasing amounts of industrial pesticides to Bt crops. The important lesson here is that pathogens and herbivores will continue to evolve new ways to circumvent the defenses of crop plants, and agriculture will continue to require the development of new forms of crop protection. If you have any questions, please let me know via the Q&A discussion board.